to 30 persons sitting here and waiting for your presentation. So you are the last today. I don't know whether this is an advantage or a disadvantage. And I ask you to be a bit shorter than 1 hour to allow some questions, if possible.
All right.
Okay.
I will do my best. So thanks, Markus, for inviting me for this school, and thanks for everybody for still being there. So I'm right now in Berlin. I just went by train to Berlin for a conference here, the first conference after a long COVID break, and I'm happy that the internet seems to be stable here from my hotel room for the moment so that I can transmit the talk here. And I'm also looking forward to meeting some of you then next week in Kyiv when I can personally join. All right, so my talk is mainly about DNA nanotechnology and why that is so fascinating for me. And I think it's a very different view on nanotechnology, and so therefore I hope it can add to your school. But first, let me start. I think we already had a speaker from Munich, and I also had the pleasure, I checked in this WUVA app, so I saw a couple of pictures that were posted there. So I have a bit of an impression of how the conference room looks. So this is how it looks in southern Germany. So I'm from Munich, down here in the very south. Can you see the pointer also?
Do we have a pointer? Yeah, we can see the pointer.
I put the laser pointer. All right. So we are here in Munich, in the southwest of Munich is our campus, and Munich, of course, is well known for its proximity to the Alps. And so here you can see the picturesque Schloss Neuschwanstein, and so we have all the lakes also around where you can go swimming, surfing, and everything around Munich. And so that is a group retreat photo from 2 weeks ago where we made a hike in the Alps, which is just like a 1 hour drive from Munich. So Munich is a big town at 1.5 million, but it does not have any high skyscrapers, or the buildings are relatively small, and so it does not give this feeling of a super huge town. And this is a bit exaggerated. The mountains are not that close, so that is a bit, I don't know, must have been a super föhn day, or I don't know how somebody took this picture, but it's still beautiful. And we have, of course, it's known for the Oktoberfest, and the beers are quite big there. And this is our campus here. It was built about 20 years ago in the southwest of Munich. So this is where chemistry and pharmacy is located, and it's a very nice environment, very modern style. And yes, we enjoy a lot working there. So I hope that some of you will get the chance at some point to visit us. We also have some newer buildings here. Like this is our newest research building for epigenetics. So it's still developing, it's still getting better. Okay, so let me introduce my personal research. So I call my group Nanobioscience. I couldn't think of a better name. And the reason it's in a way very interdisciplinary. So we have physicists, chemists, biologists. On the other hand, it is always somehow connected to single molecules, the ability to detect single molecules, mostly by fluorescence. But with that, we do a lot of different things. So one of the things that we do is we are involved in superresolution microscopy. So some of you might know that 2014, there was a Nobel Prize for superresolution microscopy. We were a bit involved in the development. And what is always important there for some of these techniques is to be able to switch off single molecules on and off. And so we worked a lot on the photophysics and the principles. How can you switch off single molecules, which is very important for that. So we did some of the basic research there. We also have a biochemistry lab. So biochemistry is very important to label molecules, to have proteins in a state that you want, and to attach maybe artificial amino acids to them that you can then put fluorescence labels. Use techniques like fluorescence, resonance, energy transfer to measure nanometer distances on protein complexes, and then to watch them, how they even move dynamically. And so we've been excited with everything to improve single molecule fluorescence, to understand the photophysics, photochemistry of that, everything that shines bright and we can follow on single molecule level. That's what's interesting us. And also we were like also in this single molecule tanks, we also try to have the coolest setups, the newest detectors, the best lasers. And that has changed a little bit. So about 10 years ago, we had a bit of a paradigm change at the group. So I came from this field of single molecule fluorescence. And that is when I met some people who use this technique, which is called DNA origami. That has changed a bit my life and my paradigm of my lab. Because we realized that this is not just playing. Well, it's fun because, well, it's a bit like playing. You have a lot of fun like you do when you play a game. But it's useful. And this DNA nanotechnology can really be used to create functions on the nanoscale. And that is what has been fascinating me since. And we combine then the fluorescence world and the DNA nanotech world to create new things. The first one was these nano rulers, really a ruler on the nanoscale that is used for super resolution microscopes. Then we went to other areas, to plasmonics, and we made single molecules very bright. I have a single molecule mirage experiment also. And also where we believe that this field is going a lot towards sensors. So we can really build very complex sensors. We can even apply forces on the nanoscale with the help of DNA nanotechnology. And I hope I can get you excited a bit about this field and tell you a little bit how that works and how that matches this context of this school. So some of the nano devices, to give you an idea, I come to first the motivation, the bigger picture. So I mentioned DNA origami. It's a form of DNA nanotechnology. So the main question that you might be wondering, why the heck would want somebody use DNA for nanotechnology? So what is special about it? So the whole point is, the first important point is forget biology. We're not talking about genetics. We're not talking about any biological thing. We're talking about a material science. So DNA from a material science perspective is a construction material. So that's what I will motivate. But before motivating, I want us to think a little bit about how do we fabricate in the moment nanometer-sized objects? How is nanotechnology actually working? We have two main approaches. Fundamentally, we can go top-down, make things always smaller, sculpture things, or we can go bottom-up from single molecules or from single atomic bonds and build in like a more chemical approach, build things. So if you think about the top-down approach, that's more the one that is successful. If you think about computer chips and everything, it's about lithography. Everything, physics is about lithography. You make your spin-coated layers, and then you create patterns with the help of photolithography, electron beam lithography. So you create patterns, you then put another layer, you do lift-off processes, and then you get final structures, which can be ultra small and ultra nicely controlled. It's of course very serial, and it's not so easy to connect this world to the world of molecules. So the ultimate dream, however, of nanotechnology, of molecular nanotechnology that was termed in the 1980s, is different. It comes really from bottom-up ideas. So the idea would be you have like this super machine, this unbelievable super machine, which is able to control single atoms. And then with the help of these single atoms, you pick one atom after the other from a depot, and then you place them, and you could assemble a little nano robot. You could create the chemical bonds that you want. You have a mechanochemistry that pushes the atom over activation potential into the place where you want them to be, and then you can build nano robot. So that is the ultimate idea, and it came from the 1980s essentially, when these scanning, tunneling, and atomic force microscopes were invented, so that people had the fantasy that if you look into the future, then you could assemble whatever you wish. And it's a bottom-up approach to controlling one atom after the other. But 30 years later, we know it has not realized to that extent. There were beautiful examples of functions, especially in quantum science, that has been achieved, but it has not become a broad technology that we can really build very complex things. So what can we do? And what is the problem maybe of this approach? The fundamental problem is this famous Smalley and Drexler dispute about it, why that could be limited, this approach. So we're in the beginning of the 2000s. So Drexler, he coined this term molecular nanotechnology, and Smalley, he was the discoverer of the fullerenes. And so Drexler is like a positive guy who was saying, oh, this will all happen and we will have atomic assemblers and we can build whatever. And Smalley is a skeptical guy and saying, well, there might be fundamental problems with this approach. And so two of these problems are the so-called fat finger and the sticky finger problem. So the fat finger is what you might experience every day on your smartphone when you type your WhatsApp message, is that your finger might be bigger than the letters that you want to hit, and then you might hit the wrong letters, at least it always happens to me. And as a consequence, you might miss the wrong one. So it's error-prone, the technology. The second problem is the so-called sticky finger problem. And the sticky finger problem also refers to the functioning of this ultimate machine, this robot that assembles the assembler. That is, that you always need a hierarchy of forces to be able to pick up an atom, so it has to stick stronger to your tool than to your depot, and then transport it with your tool to your place where it has to go, and then it has to stick stronger to the place where you are going than to your tip. And you might experience these problems when you decorate your muffins, that your chocolate streusel, that they stick to your finger and they won't go on the muffin where you want them to go because they rather stick to your finger. So that's a sticky finger problem. So there might be fundamental problems with this approach, and they create uncertainty and errors. So I'm chemist by background, so I was thinking, well, maybe one has to do it differently. We need some sort of a self-assembly process, right? And what we would need is something like an energy landscape and with some specificity. Then we could have our building blocks and just mix them in solution, and every building block would go where it is supposed to go. And maybe we can even build ways to couple this idea of nanochemistry, maybe with light, I don't know. So that could be an approach. But if you think about self-assembly, there is tons of self-assembly out there, but I still have not seen a macroscopic tool that was produced by self-assembly. On the surface, it might work nicely. You can have beautiful self-assembled monolayers, like where you have your monomers and they have some selective chemistry for a surface and they can react with the surface, and then you can get a beautiful polymer brush on a surface, perfectly ordered monolayer. That's possible. And you can also create beautiful patterns. So certain arrangement of organic molecules on certain substrate can give you lines like this, can give you hexagonal patterns, can give you nanowires, or all these beautiful different patterns. But if you think that we want to build one day a nanorobot, there's a limitation. This limitation is that these structures are super highly symmetric. I mean, a nanorobot is not like a super symmetric pattern. It could be, if you look at this pattern, that maybe I want to have an amino group here, maybe I want to bind a protein here, a nanoparticle here, a virus there, whatever. So you want something really complex and not with a high symmetry. So what you need is not only self-assembly, but you need the ability for defined symmetry breaking to really be able to place the biofunctional things as you wish them to have. Good, and this is finally now where the DNA comes into play, right? So what do you need to know about DNA? I guess many of you know the basic concepts, but very short summary. DNA is a polymer made of the phosphate backbone, the ribose, and then comes the next phosphate here from the 5' to the 3' end. So this is the backbone of the DNA strand. The DNA then carries, of course, nucleobases here, which is the guanosine, the thymine, the cytosine, and the adenine. These are the nucleobases. And of course, it's known that always two strands of DNA like to go together, right? So they form a duplex. It's always that G pairs with C, Y via 3 hydrogen bonds, and T with A via 2 hydrogen bonds. So this is here our second DNA. With this alphabet of the DNA, one can have a sequence of DNA. And if you have one molecule of DNA and you add another one, you will know where it will bind. Because here ACTG is sequence TGAC will bind exactly at this position. So you know where it will go. And this gives you now an orthogonal glue, so to speak, that you can bind certain sequences with other sequences, and only the right sequence will bind. And you can have many different sequences so that you can have many different bonds. And this is the basic idea of DNA nanotech. You have a polymer. This polymer likes to form bonds, dimerize, and these bonds will be specific, and you have an almost unlimited number of orthogonal interactions so that you can place tons of different things along this DNA strand. That's all the fundamental idea of DNA nanotech. And there's no other polymer that can do it. Maybe one day with proteins we can do that. In the moment, we do not have the right understanding for proteins that we can build such complex things. It would theoretically be possible because you have the 20 amino acid letters in the protein, but you don't have the specific interactions that it's like, so it's much more complex. So DNA in the moment is the molecule of choice that you can do such complex things. So DNA nanotech came a long way, and I make a shortcut in 2 minutes. It started in the 1980s, this idea of using DNA as a material by Nat Seaman. He's the grandfather of the field. And there was one famous breakthrough, one improvement, or one step forward that was happening in 2006 by Paul Rothemund. It's a fantastic work. It's one of the very few works I know where a Nature paper is written in the I form. So he writes, I sat down, I thought about this program, I bought the DNA, I then did the AFM imaging, and then it's a single author paper written in the I form and has a Nature cover. So already at that time it was realized it's something special. Because it was difficult, it was tedious to make DNA nanostructures before, because if you add many DNA strands that should form together, what would happen is that you form many bindings, many puzzles, so to speak, are mixed, and they start all at different positions, and then you have many half-done structures, but all of them are different and don't fit together anymore. So you had a problem of hierarchy, and that was essentially solved by Paul Rothemund. And he, in this paper, came up with many different structures. They are imaged here by atomic force microscopy. This is DNA on mica, and they are big. So the diameter here is about 17 nanometers. So this is like 19 times 17 nanometers or so. And it's not only one that worked, but if you look at the overview images, you can see that there are many structures which are more or less identical. So absolutely fantastic. One guy making many huge structures, and also in high yield. So what happened? And remember, it's DNA as a construction material. So what happened is the following. The technique DNA origami is simple. At the end, it is super simple. But what is special is that it uses one long template strand to form a hierarchy, so to speak. So this represents a DNA strand which is roughly 8,000 nucleotides long. So where the heck do you get an 8,000 nucleotide long DNA strand from? It's super simple. It's from a phage. So a phage is like a virus for bacteria. So you only have to take this virus, throw it on your bacterial culture, it will infect the bacteria, and it will produce tons of itself, so that after a couple of days, you only have to isolate your viral DNA from your cell culture, and you have tons of this. So you can really produce this in large amount, and you know the sequence of this DNA strand. So what you can do next is to buy shorter DNA strands, short so-called stapled strands. So they staple together different positions of this long strand, like here. So you know the sequences here, so you know how the complementarity works. You form this loop. So this hybridizes to these two positions, for example. This is about 14 nucleotides long. And you do not only add once of these short strands, but you start adding 2, it forms another loop, 3, 4, many, at the end, 200, about 200 of these short strands. And it forces the long strand into a predefined structure. So it's not a chance that it ends up as a rectangle, but this is how it was designed. This is the only way like that all these short strands are interconnecting correctly and are all completely hybridized. So you force this long strand into a desired shape that was designed in a computer program. So there's freeware available where you just sit there and you say what structure you want, what strand you use, and then it makes you suggestions, and with some interactions you can start creating your own shape of DNA that you would have. Then you can go further. If you look in the internet in like DNA shops, you can buy DNA in the internet, like IDT or Eurofins, and tons of companies. And they not only offer DNA, but DNA also offers a huge amount of chemistry. You can attach everything to DNA. You can buy thiolated DNA, aminolated DNA, biotinylated, click chemistry, whatever chemistry you wish, you can buy it on DNA. And you can also buy it with fluorescent dyes. So at the end of the day, you can say, I want a fluorescent dye placed right here because this here will hybridize there, and right on the other corner because this dye wants to go there. And this is what you can do. So it's all in the mixture. It all happens by itself. And at the end of the day, you don't even need to know that this is DNA. You can just look at it as a molecular breadboard where you can plug things wherever you wish. For example, light bulbs representing our fluorescent dye molecules. So we call it a molecular breadboard. And so you have the DNA double helixes, which are represented by the spray bars. You can put dyes, nanoparticles, proteins, many, many things. And it works super simple. So you use this long strand. This is called the scaffold strand. You add about 200 staples. You heat it so that all unwanted interactions are broken. And you cool it slowly. You have one gel chromatography step. And then you can, for example, put it on a mica surface, do atomic force microscopy, and you get all these perfect rectangles. And you can zoom in. You see the smallest defects, which could be also caused by the AFM tip, of course. So you see when one strand is missing, it's quite amazing. So it works so robustly, at least for this 2D DNA origami structure, that you can fill it all in one of these Eppendorf tubes. You cook your tea water, you throw your Eppendorf tube in the tea water, which is then very hot. You have your tea running, and then you drink your tea. During that time, it's cooling. And then you put it, you do the chromatography and throw it on the surface, and it's there. For these rectangular origamis, you cannot do anything wrong, almost. So this really, we did this. My students did that. They wanted to try it, and they said it works. Okay, so now the question is, of course, so that is, and maybe I don't know what their culture is. You can also ask in between, or do you wait to the end? So as you wish. I would also be fine just asking whenever the time is. So what can you do now with that? And that is the time actually when I came into this field, with meeting people like Fritz Simmel from Technical University of Munich and Tim Liedl from LMU Munich, where I'm working on. And so the first application that we did is nano rulers, and I will discuss that. Then we built fluorescence amplifiers, and I will briefly tell you what we do with those. We do other things. We have a force spectroscopy based on DNA origami, where even the origami is applying a force. It's not only measuring it, it's applying it. We're now doing some graphene biophysics, which is a new topic in the group. We think about repair and self-repair processes and such nano devices. If they work someday in your body and you have degradation and nucleases, what could you do? And how could you maybe, for example, you see it's photo-bleached, all the dyes are destroyed, how could you recover them? So that's another direction that we've been taking. And we have different labs to do that. So we have chemical lab, we do a lot of nanoparticle stuff also, and then we have our laser labs, and most of the setups are home built, so that's a bit my background where I came from. But we also had some commercial setups, and we also have molecular biology labs, cell culture, bacterial culture, all these. So it's really, everybody finds this niche what he likes most. So I want to present 2 examples of what we do with that in this course. And it's a bit driven by this idea that you don't always need a complex microscope. Maybe sometimes you can do something on the nanoscale, and that is like putting a microscope on the microscope, that you do something on the nanoscale and it creates a function that you usually would need a complex microscope. And so first example, that's essentially our long-term thing. It's our DNA origami nano rulers. Explain you a bit how that works. And then I talk a bit about our DNA origami nano antennas. And this third one I will skip. Our DNA origami fossil, it will be too long. We should rather have time for questions. All right. So actually, this time when we started that was a time when really super resolution microscopy was going crazy, around 2008-9, when we were also doing super resolution microscopy. And so instead of having actin images like this, you could now get actin images like that. So this is already 12 years old. And you went to these conferences, and everybody was showing these cool images of some cellular filaments. But with these cellular filaments, it was impossible to evaluate how good the new microscopy technique really is, because you don't have a standard to compare the same thing in different labs, right? You could not. If I have here 3 actin filaments running parallel, I zoom in and I make a cross section, and I say, oh, here you can see I can resolve 88 nanometers, so the resolution of my setup is 88 nanometers. But I mean, you cannot always search 2 days in your cells until you find a structure like you like, or that's what you maybe do, but is this representative? You also don't know how many biomolecules contributed to that, how many biomolecules were dark. How did the sample look after you measured it? Is it boiled? Is it cooked? Or what is it? So what was required, like everybody had images like that, but nobody knew which imaging technique now really is better than the others, because there was no sample to compare things. And that was a time when I talked to these DNA origami people, and we were wondering, can we place dye molecules with a DNA origami? Can we place fluorophores with nanometer accuracy? That was our question. You have like here an electronic breadboard, you can apply your electronic things. We wanted to place molecules. And then we did, well, this is not the very first we did. The very first we did with Selfassembly, so that is then a bit later with Shevchenko, who then won the Nobel Prize from Göttingen and with Leica, the company. And so they were interested because they had this head microscope and they wanted to demonstrate it with a reference sample. So we made this DNA origami. So you have a line of 12 dye molecules here, and 70 nanometers away you have another line of 12 dye molecules here. And usually that would look in a confocal microscope like that. So you scan in a confocal microscope, and then you get these spots, and one of these spots represents one of these structures. And if you look at the diameter of one of these spots, it is a diffraction limited spot. So that is the smallest you can get with conventional microscopy, which is like maybe 300, 450 nanometers diameter. But we were working with this, what is called a stimulated emission depletion microscope. And with that microscope, you can make a confocal microscope, and you can put a donut-shaped laser focus on top of the original laser focus, which would deactivate the dye molecules which are in this ring, and only in the very center the fluorescence could still be emitted. And how good that works depends on the power of this stat beam. The stronger the stat beam is, the better the resolution. So what we did is we scanned the same surface again, and you can see that now the spots become smaller because we also have the stat beam on. Now we scan it again with more power, so that the resolution is even better, and you can see that the spots get even smaller. And if you do that again with more power, what you see is that these are not single spots, but these are indeed all double spots, all of these structures. You can go further. You start here, you kind of see the line, and here you see sort of full line, and this is also because photobleaching sets it. Laser power, dye molecules degrade, they are destroyed, go into high excited states, and the bonds break or oxidize. But you can see we can measure it several times, and you can then see which is really the direct visualization of the Rayleigh criterion, the ability to distinguish two point-like light sources. All right, so that then was becoming the nanoscopic rulers. We started producing for every microscopy technique you can imagine. We started producing such rulers for localization-based super resolution, where you look at single molecules one after the others. You localize one molecule after the other. So that is the normal image, the wide field total internal reflection microscopy image. This is the super resolution microscopy image with the studded spots. And that was our first paper actually in this direction. Then this Confocal was normal microscopy. You could make very long origamis, and then you would all get the sample structures of single molecules at the end of these rods. So this you can resolve with a normal microscope. This is the longest we build. And you can make for stat microscopy, as I just showed you. You can do that for fluorescence resonance energy transfer, also the R to the -6 dependence with 50% energy transfer nanometers. So for Fred, for the three dimensions, so you can put like the splitter structures on the surface and measure the distance also in the third dimension, which is a bit more tricky than in 2 dimensions. But that also worked. And for plasmonic rulers, when you have a nanoparticle, whatever. Like the concept techniques, and up to 6 nanometers. That was at that time the world record in optical resolution, 6 nanometers. Good. So one of these techniques that works very nicely is DNA paint, which is nowadays used a lot when you localize one molecule after the other. When you have a DNA strand that binds to another strand, and it unbinds, it binds and unbinds. And if you look at the intensity of such a spot where it binds and unbinds, you can see that then it looks at it's on, and then it's off, and then it's on. It's a way to switch single molecules, and that gives you the highest resolution. And so this is an example image of a nano rulers with 3 marks at 80 nanometers distance. And you can see every of these origamis has 3 spots, and thousands of these structures. And here, many of these structures you can see within 500 nanometers. So it's really fantastic. And that was the reason why we started a spin-off company to provide this to the microscopy companies and also to the end users, that they have a sample that they can compare the things. But so that was my first application, and brings me already to my last application, which is a bit more complex. But also very fundamental. One of the things, I mentioned to you that single molecule detection is where I come from. So we were always wondering what are the fundamental limits of single molecule detection. Of course, photobleaching is one, and the other one is, of course, the brightness. The single molecule is, of course, quite dark. You need an expensive setup. You need a setup on an optical table, which might look like that. And we, of course, always want more photons. In single molecule biophysics, it can be important to get higher time resolution. The more signal you have, the higher is your time resolution. But also, if you think about single molecule applications, there are single molecule applications out there. For example, DNA sequencing by Pacific Bioscience is a fluorescence-based DNA sequence technology. There are other single molecule technologies like nanopore DNA sequencing. Super resolutions microscopies are single molecule techniques sometimes. So we're also thinking about diagnostics. And so could we do a single molecule detection instead of such a complicated setup on something that is very simple? So we need tons of more signal from our molecules that you can detect it with optics, for example, democratizing single molecule detection. So how could that work? What we need is more photons, better signal to noise, and more sensitivity. So we need an amplification mechanism. So we cannot build new cameras. Samsung or Apple can do that. We cannot do that. So we have to come from the other side. We have to make these dye molecules bright. And how could one do that? So the answer comes from nanophotonics. So nanophotonics is like everything where you process light on the structures that are smaller than diffraction limited. And one structure are metallic nanoparticles. So the metallic nanoparticles, when you shine light on it, you create, I think tomorrow morning there's another talk on plasmonics. So you polarize your particle. You have your E field, your electric field of your incoming light, and in one instant the particle is smaller than the wavelengths of light. So it will be polarized. The positive charges will go to the other side. No, other way around. The electrons will move to one side. The nuclei stay where they are. But it's polarized. And then the next instant, instant later, the light wave travels further on, and the same particle will be polarized in the other direction. So you have the always switching light field of our incoming light. And the particle reacts to that, and what is a property of noble metal particles like gold and silver is that this oscillation happens in resonance in the visible spectral range, which means these electrons will even keep on, well, there's energy built up in these particles, and they will even keep on oscillating the electrons collectively when you switch off the light, at least for a few cycles. So, and the idea of that, so the consequence of that is that these charges that are delocalized, they create their own electric field. And since you have a storage of energy in these particles, the electric fields here in these poles can be much higher than that of the instant incoming electric field of the light. So you have an up concentration of the electric field here and here on the poles. So that has been known for quite some time, and you can also do simulations of how much that is. If you have a single 80 nanometer particle, you see a few-fold intensity enhancement on the here in the poles. It's now 90 degrees turned. It gets much better when you are able to build more complex metallic structures. For example, build a dimer of nanoparticles. If you build a dimer of nanoparticles, here, this is simulated for a gap of 20 nanometers. You can already have substantial field enhancement. The smaller the gap, the more that gets. And Raman and other surface-enhanced spectroscopists will tell you it has to be super, super small, like 2 nanometers. But I'm telling you, well, if you want to do something in this gap, maybe it's better if you already achieve an effect when the gap is not so small, because we might want to use this gap. So let's think about 15 nanometers where you can have a couple hundred rests. So what happens is everything changes. The dye molecule changes its properties by coupling to this electromagnetic environment. So what happens is with the particle, the excitation rate will be higher, the radiative rate will be higher, the non-radiative rate will be higher. It's a complex interplay, not simple. And it all gets more drastic when you have 2 particles. And at the end, also the radiative and non-radiative rate from the triplets get higher so that you can also increase the photostability. The fundamental problem is, of course, how to build such a structure. You can do top-down lithography again. Yeah, so nanolithography, it works. There's beautiful work on that. But what nanolithography is not good in is to place molecules, single emitters in these gaps. That's what lithography is not good at. It's good at making structures, although it's also difficult, but it's not good at placing single emitters in the right place. How could you do that? And this is how to place molecules or molecular assays in the plasmonic hotspot. That's the key question, and that's what we do with the help of DNA origami, of course. So we build this pillar-like structure of origami, and we find nanoparticles there. They are also bound by hybridization of DNA. Like here's a particle surface, DNA is coming out of it, forming a duplex with DNA that's coming out of this gray DNA origami structure. That's how it works. Then we can place a dye molecule there. As before, we shine light. We hope that we get a lot of signal. And this is how it looks. So you have single molecule fluorescence. This is the intensity, really photons per 10 milliseconds that we detect, like 100 photons per 10 milliseconds. A typical single molecule signal that will work for a long time. And what you see, it goes on and off. And this on and off blinking can be a triplet state. Here in this case, it is a radical ion state. So the for a moment, it loses an electron and then it gets it back. It loses it to the environment. And this is also proof that we have a single molecule, right? So if you have many molecules, you would not see if a single molecule is off or not. But if it's a single molecule, the signal goes from 100 to 0, right? So that is why we know it's a single molecule. And then it photobleaches here. This is a moment where you have a photoreaction and the dye is gone. It happens in every single molecule fluorescence experiment. We zoom out. And this is our signal we worked with for 20 years. And now we're at the particles. And you can see with a single particle, the signal goes about, increases about 8-fold. And you see the molecules are still blinking, bleaching. So that's an example trace of a single molecule. And if you add both particles, you have an increase of another factor of 10 roughly. And you still see blinking. It seems to be bleaching quicker. But if you think about the brightness, so the number of photons, which is like the integral, that is increasing. And this is important. Brightness is important, how many photons per time, and then the overall dose is increased. So the next question for us was, can you do something with that? Can this become somehow useful? And that's why I meant we don't want to have this hotspot too small. So we were thinking of making a DNA hairpin assay. And so you have a dye molecule and you have a quencher of this hairpin. And then if your DNA or RNA of a virus comes in and you want to detect a virus, so this was for Zika virus, then the incoming target DNA, it would bind with this hairpin, it would open this loop, and then this dark quencher would be displaced from the dye and it would become bright. And this we wanted to build into our nano antenna. And we could see that it's opening for an RNA target, which is for the Zika virus, a similar DNA target. So we see about 40-50% opening, which is okay. We have a bit of unspecific signal. We later found out that that is because this one is also bleaching, this dark quencher. But the fluorescence enhancement was disappointing. We only had a factor of 4- or 5-fold of fluorescence enhancement. So it was not getting very bright. And when we analyzed it further, what we realized is that in fact we did not have these dimer antennas, but only one particle would bind. Because this assay is too bulky, it was taking too much space, so that the second particle would not bind, and then we would not get enough. So that was a bit disappointing, and so we said, okay, can we do better? And the problem, of course, in this construct is, it's here that the DNA is in the middle. This is where the hotspot is, this rod is right where the best space is. So we cannot use the best space, so to speak, for the assay because the DNA rod is there of this origami. So we created a completely new DNA origami, which you already saw in the one graph. And this is like this twin tower design. And here's where the hotspot is going to be. So this from the other side, it looks the same, but if you turn it, it looks like this. And this, now the particles come from the front and from the back. So it's behind the binding site and in front. This is where the two particles will bind, and this is electron microscopy image. This is an averaged image where you can see that. And so we call this nano antennas with cleared hotspots, nachos, and hope that now here we have more space that we can use for biomolecular assays. And we even managed to get an electron microscopy image of the fully assembled nano antenna. Here's the origami. Here are the particles. It looks really like our drawing. We were amazed when we saw that. So this is our data from 2012. So the question is, does this structure work? And if we compare the data, we see it's even quite a bit improved by a factor of 3, 4, or 5. So, and that's because the gap, we made the gap a few nanometers smaller. Right. A bit of heterogeneity still in the structure, but the molecules get really, really bright. Okay, so can you now do something with that? And we now made a different assay because before we also had this problem that this dark quencher would be photobleaching. So we have our capturing strand, and comes the target, which in this case was for antibiotic resistance. It would bind here in the hotspot region, and it would be a bit longer. The target is a bit longer, so that then, after it bound, we could also bind images strands, which carry a fluorescent dye, so that then the images strand here would be fluorescent and could be enhanced. And that's how it works. So this is, we have a green dye in the origami down here, that we know where the DNA origami is, where the nano antennas are on the surface. And then we do the assay and we see that it works in buffer and even in heat deactivated serum. We find pretty much the same sort of signals. And then we can see if we don't have the target, we essentially have almost no false positive. And we can see that the binding yield also is very much the same if we have the nanoparticles or if we don't have the particles. And without the target, we essentially also have almost no false positives, except when we don't have the nanoparticles. It seems that the nanoparticle DNA, the DNA that binds the nanoparticle, creates a bit of unspecific binding to the target. All right, and then we can also study the enhancement. And fortunately now we still see our enhancement of a couple hundredfold or even sometimes a bit more. So we have a lot of enhancement. It works. We can distinguish really, these are single molecules without the nanoparticle. And this is only the beginning of the distribution that you see here to show that we can completely distinguish these two when we have specific binding in the hotspot or not specific or in nanoparticles. And this works in serum and in normal buffer. So there are bleaching step analysis and so on that we know that's more. So bringing me to the last subtopic, which is what can we now do with that? Because it gets much brighter fluorescent molecules, and also not only brighter, but also we can do single molecule assays with that. So the fundamental idea is, well, what we saw in the literature at that point is an article by this lab from Aydogan from UCLA, and they claimed that they can detect 200 molecules on a smartphone, 200 fluorescent proteins on a virus. And so they have this portable cell phone setup where you click your smartphone into this device, which has a little laser diode, and they claimed we can detect 200 molecules. So we said, if they can detect 200 molecules, we might be able to see single molecules. So that's what we tried to do then. And there was a perspective article by Michel Ry, one of the first guys in the world to see a single molecule. And so he made this Photoshop image. So that would be the vision to detect single molecules like that. So can we do that? And this is what we did. So this is now our real, it's not handheld, but it's portable microscope. So we have our laser coming from here. Here's our sample. We have an €8 objective lens from the internet. It costs almost nothing. The filter that we use is a normal filter, costs a couple €100. And then here's a smartphone on top. So it's our focusing screw. And it's battery driven. So we have a little battery pack for that. And this is then a photograph from the top. You see these spots. And what you can then see, look at these spots here. You see they really disappear in a single event. Sometimes they blink a bit. This one now disappears, not this. And then finally, this one also disappears, this very bright one. So you can see, bam, they go away in a single step. And this is, of course, a sign that these are really single molecules. This is raw data, essentially. And from this, you can extract now single molecule trains in from these movies, and you see that they look pretty much the same. Single step photo bleaching. This is the single molecules in the antenna. This is the assay where we detect single DNA. So we have 3 binding sites. You can have up to 3 bleaching steps. Here we really detected single DNA molecules so single molecule detection on a portable smartphone microscope possible. Brings me to my summary. I hope I could get you excited a bit about DNA nanotech and what you all can do with that. I showed only a couple examples. It's a huge field out of there who try to change the world and try to find the big application. I showed you a bit the DNA origami nano antenna, which is now summarized in this review, our nano ruler approach that is even commercialized. I did not talk about the graphene superresolution. That's the high-end super resolution that we are currently working on. This is our group from last year's summer. The unofficial picture, this is the official picture, of course. And I would like to thank our cooperation partners. The nano antennas were for many years developed with our junior group on Punya for the nanoparticles. The others are not here for our other projects. This is my current group. I thank these funding organizations. I thank you for your attention, and I hope that you could follow and that it was good to understand over there, and so that you could ask. Thank you so much, and hope to see you soon.
So, Philipp, thank you very much for your presentation. I think the auditory is open for questions. Oh? He has left already?
No, who is left? I haven't left.
No, because you didn't say anything.
Oh, there was a question. I could not hear it. Sorry.
So,
I didn't.
Questions from the auditory, please. I think the auditory is tired.
Yeah.
I can understand. It's only me now between you and dinner.
So it's not clear. It's not only clear. Not in the best. It looks like on the side.
What? No, this is
It's presentation.
this is the presentation. Okay, so if there is no questions, thank you, Philipp, and I really look forward to hear your presentation next week in Kyiv in person, because in between I had to leave the room for some other discussion, so I only got the first half of your presentation. So thank you again for sharing your knowledge with us online, and then
Yes, thank you.
well, give him an
Thanks, and have a great time there, and hope to meet some of you then next week. All right.
thank you. Bye bye then.
Bye bye.
Thank you.